Okay, so part three, now we're going to talk about why different populations have allele frequencies. So I kind of asserted earlier in this lecture that different populations have different allele frequencies. This is very useful for identifying ancestry, but what is the reason why they do? And so now we have to talk about evolution. And so there's a couple of evolutionary forces that are thought to be really important for this. So natural selection. So this is the process by which some sort of phenotype is at an advantage. And so that phenotype spreads in a population because those individuals are more likely to survive. That phenotype is controlled by some sort of allele, and then that allele spreads in the population. Genetic drift. This is just a random process. Migration, when individuals from one population go to another. And then finally bottlenecks, when you have either a huge reduction in the number of individuals in a population or founders that found a brand new part of the world. So natural selection, so this is a major force driving allele frequency change. Chief mechanism for transforming populations, and it's thought to be the principal force that shifts allele frequencies within large populations. So in general, if you see a large shift in allele frequency from something like 10% to 80%, most likely that is caused by natural selection, at least in a large population. So if you have a small population, that might not be true. And so this was discovered independently by Charles Darwin and Alfred Russel Wallace. And the concept of natural selection is pretty straightforward. I think you should probably understand it by this point pretty well, but individuals exhibit variations in phenotype. So this could be something like height, and some variations are heritable. So height is an example of a very heritable trait. So a lot of your height is determined by genetics. And so genetic differences account for some sort of change in phenotype. More offspring are produced than can survive. So if you can have, this is not really true anymore of humans, but for most populations this is true, where there's a carrying capacity where the resources can only support, let's say 100 individuals. So if you're a wolf, if you have too many wolves, eventually there's not enough deer or other things to eat, and so you end up starving. So there's this idea that there's a limited number of individuals that can survive each generation, but typically, you don't produce 100 each generation, you produce something like 200 each generation. 100 of those offspring end up dying. And so you end up having this opportunity for some phenotypes to be at an advantage against other phenotypes. And so some phenotypes are more successful and reproduce at higher rates or have higher survivability, and then that can change the allele frequency based upon those phenotypes that are changing. So in humans, there's actually some really cool examples of positive selection that have recently acted, and one of the first ones that was discovered, and still a very powerful example of positive selection, is lactase persistence. And so lactase persistence is the ability to use milk as a food source in adults. So milk is something that all mammals produce, but typically milk is only used to feed offspring when they are young. Humans are very unique in that they collect milk from other animals to use as a food source in adult individuals. And so milk has become a very important part of human diet. But mammals in general and the human ancestors cannot actually metabolize milk as an adult. And so the reason for that is that they lack this enzyme called lactase, so human lactase. And this is an enzyme that actually metabolizes the primary sugar in milk. So lactose is a sugar. It's composed of it's a disaccharide, so it's a covalent bond between glucose and galactose, and this is the primary sugar in milk. So mammalian offspring, when they are young, they produce this lactase in their gut, but as they become older, this lactase enzyme is turned off, and adult individuals no longer produce this enzyme. And this is also true of human ancestors. So human ancestors were unable to produce lactase as adults. And lactase is important because it's actually required for the metabolism of lactose. And what it does is it breaks up lactose into glucose and galactose. And then these enter metabolism and are converted to energy like ATP. So what happens if you drink milk and you're an adult individual? So if you have lactase acts in the small intestine. The lactose is converted to glucose and galactose, and the glucose and galactose are taken up by your small intestine and used for metabolism. If you do not have lactase, the lactose ends up passing through your small intestine into your large intestine. And so this actually causes a number of digestional issues. So one reason is that you have effectively an osmotic shock, because you have so much of this lactose sugar in your large intestine that causes osmotic shock that causes diarrhea. And then a secondary problem is that because you are unable to metabolize the lactose, there is a large sugar source for bacteria that live in your large intestine. So lactic acid bacteria are actually able to ferment the lactose, and as a result end up producing a large amount of methane. And so fermentation produces a large amount of methane, which causes flatulence, bloating, and constipation. And so because these bacteria have this rich food source, they're able to multiply, and then they produce all of these byproducts in your large intestine that are problematic for your digestion. So if you don't have lactase, you end up being very uncomfortable if you drink milk. So recently, humans have evolved the ability to metabolize milk as adults, and this phenotype is called lactase persistence. And so it's just simply the fact that we produce lactase as adults. So there's been a change in the promoter that allows us to use milk as a food source. And so, you always have to be a little bit suspicious of kind of evolutionary arguments. That's one of the research focuses of our lab here at Georgia Tech. But what is thought is that the ability to use milk as a food source somehow was advantageous to individuals that could process milk. So perhaps in periods of starvation, they were able to drink milk. Perhaps milk is a easier food source in order to farm, to have cows that kind of just produce milk, and then instead of having to sacrifice the cows and eating the meat, you can simply just keep the cow alive and produce milk. We don't really know the exact detail of why it's advantageous, but the fact that it has spread provides really strong evidence that these alleles are under positive selection. And so we can see this, and we can see this by looking at different alleles that provide lactase persistence. So there's actually not just a single allele that has arose in human populations, but there's actually different ones. And so these are named by their position in the lactase promoter. So you've had a change to a G at the 13-907. 3 nucleotides later, there's a T. But kind of what all of these different mutations do is they allow the lactase allele to persist in adult individuals. But you can see that lactase persistence has evolved independently in different parts of the world. So if you're European and you're able to drink milk, most likely the reason for that is the prevalence of this allele right here. If you're from the Middle East, most likely your ability to process milk is from this allele right here. If you're from Africa, there are 3 different alleles that have spread in different parts of Africa. There isn't a lot of lactase persistence in Asia, especially historically, although these alleles can now, because of the spread, because of travel and kind of migration, these alleles can now spread to Asian populations as well. So this is an example of how you've changed the allele frequency of these specific alleles. But you're only changing alleles within this gene. You're not actually changing allele frequencies outside of that, to a large extent. Pathogens are also thought to provide an important source of natural selection, and pathogens live differently in different parts of the world. So depending on where you live, the environment is very different. So this can be something like climate, how hot it is, how cold it is. This can be density, so you can also have differences in cities. And so, some pathogens are more likely to spread in dense populations than non-dense populations. Pathogens spread through different vectors, so mosquitoes, rats, fleas. So there's a lot of variation in pathogens and where they live in different parts of the world. And so this can also have a very important part of natural selection. And you can see this, for example, in salmonella susceptibility. So this allele right here, this is just a way to name an allele. This is associated with resistance to salmonella. So you're less likely to die of a salmonella infection if you have this derived allele. And so you can see that this allele is much more frequent in these parts of the world, where salmonella is much more common and a much stronger source of fatality kind of in these different regions. So pathogens are another very common source of natural selection. So we talked about diet here. Now we're talking about pathogens, and kind of one final example that's a pretty cool example is adaptation to high altitudes. So the higher altitude you are, the lower oxygen concentrations there are. And so it becomes harder to breathe. For an example of this is Mount Everest. If you try to climb Mount Everest, there's a huge fatality rate, and a large part of that is the lack of oxygen and the lower oxygen in the atmosphere because you're going so high. It's known, for example, that Tibetans who live near kind of the Mount Everest mountain range and very high altitudes are much more adapted to these reduced oxygen concentrations. They're much less likely to suffer from altitude sickness, for example. And so, researchers have tried to understand the basis of this phenotype. And so what they did is they compared two populations that were very close to each other. So the Tibetans that lived at high altitude, and then nearby Han Chinese, not nearby, but somewhat nearby, that are thought to share similar ancestries from Beijing. And they compared the allele frequencies between these populations. And so you can see that there are a small number of alleles that have a huge difference in frequency. So in the Tibetan populations, approximately 80% of the allele frequency is approximately 80%, and then in the Han Chinese population, it's only about 10%. And so this is actually a gene called EPOS1, and this is a hypoxia-induced transcription factor. And so this transcription factor is activated when you're in low oxygen levels. And so this changes the expression. This is thought to change the expression of it kind of in some sort of way. And we'll get to this in a second, but this haplotype actually comes from an ancestral human population called Denisovans. So this is also something that we're going to start to see over and over again. So this is how natural selection can affect allele frequencies. Migration is another way. The idea is that you start with populations that have different allele frequencies, but then individuals migrate away from it. So an example of this is, if you had a large migration of Tibetans to different part of the world, then you would start to increase this EPOS1 allele in those other populations. If individuals that are lactase persistence, if you have a large migration from one part of the world to another, then if it's kind of like a one-way migration where this individuals are constantly going to this part of the world, then you're going to slowly increase the amount of lactase persistence and this allele as more and more individuals go there. One kind of cool example of this is looking at the Y chromosomes, which is very common in Mongolia. And this Y chromosome is actually thought to be a legacy of Genghis Khan. So this is the Y chromosome that Genghis Khan is thought to have. Genghis Khan had a large number of offspring. His offspring also had a large number of offspring. And so as a result, there is a large number of individuals in Mongolia that have the same Y chromosome as Genghis Khan. And then these individuals ended up migrating to different parts of the world. And so you see this increase in different populations of individuals that share that Y chromosome. And so this is an example of migration that can cause such a thing. Genetic drift. So genetic drift is another approach that changes the allele frequencies. And this is due to just random fluctuations. So because populations are not infinitely sized, you only pick a certain number of individuals to seed the next population. So if you start with something that's 50-50, and then you randomly just draw 10 alleles to seed the next generation, you might not get exactly 5-5. You might get 6-4. And so that changes the allele frequency in the next generation. And that can happen each generation, where the next one it's 7-3, 4-6, etc. So genetic drift can kind of go in both directions. But over time, you can end up having a change in allele frequency because of that. And then the final thing that we're going to cover is population bottlenecks. And so the idea of this is that you have a large initial population, but for some reason it goes through a large bottleneck. And so there's a couple reasons that you can have that. Either you have some sort of thing like a drought. So instead of being able to support 1,000 individuals in a location, you can now only support 10 or 100 individuals. And so you go from a large population to a very small population. And so what happens is because of the low numbers, again, it's very similar to genetic drift, where just by chance you end up having a change in allele frequency, but you end up having kind of this permanent effect on it. And this is also common in founder effects. So when you have a population that lives in one area and a certain number of people migrate to a brand new area where that organism doesn't exist, then you end up having essentially a bottleneck. And so a great example of this is comparing African genomes to non-African genomes, and that is because non-African genomes, Europeans, Asians, etc., North American, Native Americans, all of these individuals ended up coming out of Africa. But the African population was very large, while the founders that left Africa to eventually seed the rest of the world were kind of initially very small populations. And so if you compare the amount of genetic variation, there's much more genetic variation in Africa than there is in the rest of the world, and that is thought to be due to founder effects. All right, so those are the evolutionary forces that I expect you to know that can affect allele frequencies.